It's really my pleasure to introduce Doris Adao. And I think back to when, how this work actually started when Craig and I were sitting next to one another, Craig Simmons, at one of these Ted Roger events, and we started talking about this, and this is where a cardiologist and an engineer sat down more than 5 years ago, I want to say closer to 10, and put in grants and got funding, and then this got started. So the engineer, the basic scientists, and both of us, and the cardiologist really, gave birth to this. Doris has been incredible for us both. She is a PhD candidate in the IBME program at the University of Toronto, co-supervised by myself and Craig Simmons, and she will be presenting her work on investigating the role of microRNAs in hypertrophic cardiomyopathy. Doris.
Thank you, Dr. Belia, for that introduction. Good morning, everyone. Once again, my name is Doris Adao. I'm a PhD student in the Institute of Biomedical Engineering, and today I'm excited to talk to you about our work on modulating hypertrophy and contractility using microRNAs in an iPSC cardiomyocyte model of hypertrophic cardiomyopathy. Just a brief disclosure, I'm a research scientist at a startup company, Boutique Solutions, and we do have a cardiac product. To begin, hypertrophic cardiomyopathy, or HCM, is an inherited disease involving hypertrophy or thickening of the heart muscle. And clinically, this is characterized by a left ventricular wall thickness that's greater than or equal to 15 millimeters, without overt causes such as hypertension or changes in loading condition. It is the most common cause of sudden cardiac death among young adults and athletes, and provides a really wide range of expression, including individuals who are relatively asymptomatic and benign, all the way to really aggressive forms with high risks of cardiac failure and sudden cardiac death. As Dr. Chen mentioned earlier, HCM is also one of the most common genetic disorders in the world, and it's really mutations in several cardiac structural proteins that have been identified as the cause of this disease. Overall, it's estimated that over 50% of all patients with familial HCM have mutations in either the MYBPC3 gene, which encodes for cardiac myosin binding protein C, or the MYH7 gene, which encodes for beta myosin heavy chain. And both these mutations have led to phenotypes such as contractile dysfunction and hypertrophy. Pathways that coordinate the hypertrophic response are well established in the literature. However, one of the things that are recently emerging are these things that could potentially modulate these pathways, which are microRNAs. And microRNAs are small non-coding RNAs that modulate protein translation at the post-transcriptional level. And so what these microRNAs will do is they will bind to target messenger RNA and either repress its translation into protein or destabilize it and incite degradation. And so my objective for the study was really to identify and target differentially expressed microRNAs in HCM in hopes of mitigating the progression of this disease. And to do this, we were able to obtain MYBPC3 and MYH7 disease iPSCs with the healthy family members as their control, who are genotype and phenotype negative, differentiate them into cardiomyocytes, and isolate total RNA and send off for a microarray analysis. In order to validate what we were finding in vitro, we were also lucky enough to obtain MYBPC3 and MYH7 disease cardiac myectomy tissues, as well as control heart tissues, and send them off for microarray analysis as well. For our microarray results, we were able to identify several significantly differentially expressed microRNAs, both in the iPSC cardiomyocytes and the tissues. But what we were particularly interested in, as you can see here in panels E and F, are these microRNAs that are being significantly differentially expressed both in the iPSC cardiomyocytes and the myectomy tissues in the same fold direction, meaning they're being both downregulated or upregulated both in the iPSCs and our myectomy samples. And so going forward with these microRNA clusters that we've identified, we went into MERTAR-based to look at genes that have been experimentally validated as their targets using strong experimental experiments such as reporter assays, Western blots, and qPCR. And from there, using these gene targets, we went on to gProfiler to look at what these microRNAs are specifically targeting. And so for the MYBPC3 microRNA clusters, we saw that they were targeting pathways related to cell proliferation and growth, apoptosis, as well as inflammation. And when we looked at the MYH7 microRNA clusters, we also saw that they were targeting cell proliferation and growth, apoptosis, as well as oxidative stress signaling. But what we found really interesting was that in both MYBPC3 and the MYH7 microRNA clusters, EGFR tyrosine kinase inhibitor resistance, or EGFR TKI resistance pathways, was consistently being named as one of the top hits. And so we decided to look at a deeper delve into the literature and identified miR-95P as actually the top microRNA that was regulating these genes in the EGFR TKI resistance pathway highlighted here in blue. And so from there, we validated that miR-95P is indeed downregulated in our disease samples and went on to do more experiments with transfection in order to see what the role of miR-95P is exactly in these mutant cells. And so we took microRNA mimics, which would significantly increase the expression of microRNAs, or miR-95P integramers, which would decrease the expression of miR-95P, transfected them into our iPSC lines, and assess the differences in terms of the disease phenotypes once the transfection occurred. And indeed, when we increase the expression of miR-95P by treating mutant iPSC cardiomyocytes with miR-95P mimics, it resulted in a significant decrease in cardiomyocyte size, suggesting that increasing miR-95P expression could potentially reverse the hypertrophy that's happening in these cells. What was even more astonishing is that when we decreased the expression of miR-95P by then transfecting miR-95P integramers, it resulted in a significant increase in cardiomyocyte size in healthy control iPSC cardiomyocytes. And again, this suggests that now decreasing miR-95P expression could potentially be what's activating this pathological hypertrophy response in these cells. When we also looked at contractility using particle image velocimetry, we saw that treatment of miR-95P mimics significantly decreased the contraction in mutant iPSCs. So you can see here there's a decrease in the magnitude in the MYBPC3 mutant iPSCs once we transfected them with the mimics. And when we also looked at miR-95P mimics and mutant iPSCs, it caused a significant decrease in total contraction time and the time from baseline to peak in the MYBPC3 iPSC mutants, as well as modulated the time from peak to baseline in healthy control lines, suggesting that miR-95P has a role in various contractile metrics. And so to conclude, miR-95P is significantly downregulated in mutant iPSC cardiomyocytes and myectomy tissues, and had been shown in literature to regulate the EGFR TKI resistance pathway, which are involved in processes such as cell growth and proliferation. And we're currently doing studies to look at the mechanistic insights in this particular pathway. We also saw that increasing miR-95P expression using mimics significantly decreased cardiomyocyte size and reduced contractility, magnitude, and time in mutant iPSC cardiomyocytes, while decreasing the expression of miR-95P using integramers induced hypertrophy in healthy control iPSC cardiomyocytes. Therefore, miR-95P could potentially be a target for mitigating hypertrophy and hypercontractility, which is observed in HCM. And with that, once again, I'd like to thank my supervisors, Dr. Billy and Dr. Simmons, as well as my committee members, Dr. Fish and Dr. Vasconcelos, and all the members of the Billy and the Simmons lab. Thank you.